Hello everyone, welcome to a lab science flow cytometry classroom. During the flow cytometry experiment, we need to prepare the test sample. Meanwhile, different control groups also need to be set up. The previous video has introduced blank control and single positive control. Now, let's talk about isotype control. Isotype control refers to having the same species origin, subtype, fluorochrome markers, dosage and concentration as the flow cytometry antibody used, but has no specific binding to the target. Setting up isotype controls is usually used to eliminate background staining caused by the non-specific binding of antibodies to antigens. The background staining that cause non-specific binding formation usually has multiple factors. Firstly, in flow cytometry experiments, the binding of antibodies to markers is accomplished through the specific binding of the Fab terminal of the antibody, while the surface of white blood cells except T cells mostly expresses Fc receptors such as CD16 and CD32. These receptors can non-specifically bind to the Fc terminals of various antibodies, resulting in non-specific staining. In addition, in the surface staining experiments of living cells, both monocytes and macrophages have phagocytic antibodies or non-specific binding with fluorochrome dyes. In multicolor flow cytometry experiments, there are also the influences of fluorescence background and drug treatment. When these situations occur, the autofluorescence caused by the above influences can be excluded based on the experimental results of the isotype control, which helps to obtain more accurate experimental results. Therefore, if only the blank control is used to circle the gate during the experiment, some false positive results may occur. If the positive and negative are distinguished by referring to the isotype control, the false positive caused by this part of non-specific binding can be excluded. So, in what circumstances is isotype control necessary? Firstly, for markers with continuous expression and unclear positive and negative groups, such as activation indicators like CD25, CD69, and CD38 as well as cytokines like IFN-gamma, IL-4, and IL-17A, isotype controls need to be set up. In addition, when monocytes are stained with antibodies labeled with tandem dyes such as PE-cyanin-7 and APC-cyanin-7, it is very necessary to use isotype controls. This is because monocytes generally tend to non-specifically bind to dyes such as cyanin-7. In this situation, set isotype control group is helpful to obtain more accurate experimental results. When the expression of antigens is unclear, it is also essential to use isotype control to define the negative cell population. Finally, the expression levels of intracellular factor markers are relatively low, and the intracellular staining process is also very likely to lead to non-specific binding of antigens and antibodies. At this time, the experimental results of isotype controls are also needed to help eliminate the influence. That's a wrap for this video. Stay tuned for a lab science, and let's keep pushing the boundaries of discovery together.